Good afternoon. A warm welcome to this edition of the Science Breaks event. I'm Chu-Fan Lee, a senior lecturer in the Department of Bioengineering at Imperial College, and I will be chairing this event. It is my great pleasure to introduce today's speaker, Professor Robert Endres from the Department of Life Sciences. Robert obtained his master's in physics from the University of Göttingen in Germany and his PhD from the University of California at Davis. He was then a postdoctoral researcher at Princeton University. Robert came to Imperial College in 2007, and he is currently Professor of Systems Biology and leads a research group that tackles fundamental problems in sensing and signaling. Robert is, of course, a world-leading scientist in this subject area, and he was elected a Fellow of the American Physical Society for his excellent work. Besides his scientific excellence, I would also like to highlight Robert's leadership in promoting the exciting field of biological physics. Robert is currently the Director of Physics of Life, a network of excellence at Imperial College. And nationally, his effort in promoting biological physics was recognized in 2018 when he was awarded the Tom Duke Prize by the Biological Physics Group at the Institute of Physics in the UK. Now, Robert's talk today is how cells perceive their environment, lessons from bacteria. I'm sure we are all looking forward to the presentation, but before I pass over to Robert, I would like to remind the audience that you can submit questions at any point using the Q&A at the side of the screen. And this event is being recorded, so if you do not want your name to be read out with your questions, then you can submit questions anonymously. And if you like a question that someone else has asked on the Q&A, you can give it a thumbs up, and then we will try and make sure the most popular questions are asked first. Now, without much further ado, I will pass over to Robert.
All right. Can you see and hear me?
Yes.
Thank you very much.
Yes, Robert, we can also see your taskbar, unfortunately, on your slides. Are you able to lose the taskbar and make it go full screen? That's brilliant. Thank you. That's it. Done it. Thank you.
Okay, thank you very much. Thank you very much, Chu-Fan, for the kind introduction, and good afternoon to everyone else. So, as Chu-Fan said, I have a background in theoretical physics, and quite early on in my career, I went towards biology, and 20 years later, I give this talk about how cells perceive the environment, lessons from bacteria. So, what is it about? I mean, bacteria are tiny, and what I'm showing here is a bacterium which is, let's say, magnified by 1,000,000 times. And what I'm going to talk about is how these bacteria sense chemicals in the environment, and then how they signal is transduced to regulate the flagella, which leads to motility in these bacteria. So why do we care about bacteria? Because bacteria are everywhere. And they don't really live in just in simple petri dishes. They live in complex, inaccessible environments generally. So on the left side, you see multicellular communities or biofilms where these bacteria live in. And they are really everywhere in our body. In particular, in our intestine, which is shown here, we have roughly 2 to 3 kilograms of bacteria. And they sit here in the mucus layer, and they help us digest food, and they keep our immune system sharp. Interfacing here by the epithelial cells, and then of course they're also important for disease development, and that's also why we eat our prebiotic yogurts to keep these bacteria we very much like happy. In fact, we have 10 times more bacteria cells, so many more bacteria cells than human cells. We really are a superorganism. We're really an ecosystem of bacteria and viruses and all kinds of stuff. So that's why it's really important to learn what these bacteria sense and what their microenvironments might look like. So here I'm going to talk mostly about E. coli and how they swim is. They have these flagella, these appendices, and when the motors which rotates them go in one direction, then the cell runs. And if one of the motors goes in the opposite direction, these flagella fly apart, and then the cell tumbles and finds a new direction. So what happens now is it can do biased random walk, meaning it has it suppresses tumbles, reorientations when it moves in the right direction. And so on average, it moves towards the chemical sources like nutrients. Here on the right side, there's also an electromicrograph of a motor, and there are other motility mechanisms, but E. coli, that's how E. coli moves. And here what I'm going to talk about is, I'll talk about the remarkable signaling properties these cells have, and this is in principle shared with other cells as well, and in particular what we can learn about the environment. So one of the remarkable things is that they can sense very small amounts of ligands of chemicals in the environment, and this is then amplified here in terms of internal activity, and then is used to regulate the motors and the swimming. And besides that, cells can adapt precisely. So this activity, let's say, has a particular value, and when you stimulate the cells persistently, you do get a transient response, of course, but then even with the stimulus on it, it adapts back to the original level. And this adaptation mechanisms then allows also the cells to work over a wide dynamic range of ligand concentrations. And the second thing I wanted to briefly touch on is how are these cellular resources allocated towards chemotaxis? So you can think of the proteins in a cell, in a finite cell, as this pie, and if one slice of pie is bigger, then other slices of pie have to be smaller. So let's say the cell finds the chemicals and takes them up. They could now be invested in making more ribosomes and investing in production of proteins and cell growth, or it could also make metabolic enzymes to take up more, or it can be invested in other things like cis fractions or chemotaxis. And chemotaxis is very costly, as you can imagine, based on these very long flagella. So how are these resources allocated, and is there maybe some cost-benefit trade-off? So now, of course, we could go into all kinds of details. So there are decades of research in biochemistry and genetics and so on, but I want to spare you the details because it's very, very complicated. Of course, the metabolic pathway one could look at, but we glance only over it. Signaling is very complicated. Of course, there are many other signaling pathways in cells, not only chemotaxis. And then you could talk on a slower time scale about gene regulation, and this is horrendously complicated as well. So we skip all this and effectively cut to the chase. So here I want to talk about something historic. Even the time before genetics and also biochemical details were known, one could still do very cool cell biology by just stimulating cells and then looking at the output and inferring from the output what might be happening inside cells. So a classic assay is the tethered cell assay, which was heavily used by Howard Burke. And what happens here is you can attach cells with their flagella to the glass slide, shortened flagella, and then you can essentially study the behavior of a single motor. It can go counterclockwise, and if all the motors would go counterclockwise, that would mean the cell is swimming or running, or it could go clockwise, and that would lead to tumbling if the cell would be swimming. And then, of course, you can look at many, many cells and under the microscope, as was shown here. Of course, when you look at a single cell, it switches binary between these two states, and it's very noisy and so on. However, when you average over many cells in this field of view, you would get quite a smooth curve. And of course, it's a counterclockwise bias, a sort of probability per time to be in the counterclockwise state. And then you can actually learn a lot just from the output you can measure. And this is sort of a classic experiment. So averaged over many cells, the cells have some value of counterclockwise bias, and then you can apply a very short ligand pulse, sub-second, and then you see a nice period afterwards where the cells run more on average. There's an undershoot, so now they tumble a bit more, and then it goes back to the original level. This is called impulse response. So one can immediately read a lot out of this plot. So the initial spike, you can infer the amplification or gain, which is roughly 50. Then you can immediately see that the response takes several seconds. So the memory effectively in a cell is several seconds long. You can look at the areas above and below, and they are actually equal, which is a signature of precise adaptation. Furthermore, you can see that the cell weighs the near past versus the long distant past differently with different signs relative to the baseline, and effectively it allows the cells to take time derivative measurements. So the cell can make changes in time measure these. If the cell swims with some velocity v, it can infer indirectly the gradient in space. Of course, now we know much more about the chemotaxis pathway with structural biology and genetics, of course, highly developed. And so how does it work now? So if the cell swims in the wrong direction and less is binding to the receptors here, and since the activity of these downstream molecules is very high and there's some phosphotransfer, and these molecules can diffuse and bind the motor. And that leads to clockwise rotation, which means tumbles in, and the cell wants to reorient. If the cell swims now up the gradient, of course, there's more binding now, and then you don't have the activity. And then you don't have this guy, you have this guy instead, unphosphorylated and no binding. And then the motor is in the default state of counterclockwise, meaning the cell keeps running in the right direction up the gradient. There are also adaptation enzymes, these KRKB, which extends the dynamic range, and this is, of course, this adaptation mechanism. Now, of course, we want to have more quantitative experiments, what's happening inside cell, and my collaborator Victor Sercik develops this really nice method in these bacteria, which is known as FRET. And so what you have is here, you have a laser and you shine it on the cells, and this is genetically modified to proteins. If they're close together because of activity, there's energy transfer and you emit again at a different wavelength, you can infer the concentration of these pairs and the activity indirectly. And you can see what's happening inside cells. Then you can understand to a step change that the adaptations the cells show is highly precise, which is shown here. You can also look at different adaptation states, cells with different adaptation states, and how the activity is as a function of ligand concentration. And you see immediately this wide dynamic range over which these cells can respond, or the receptors. And from this sigmoidal shapes, the steepness, you can infer how cooperative these receptors interact, and so it leads to the gain. And in fact, you know, when you have a ligand coming in here, it doesn't only change the conformation of one receptor, it changes the neighboring receptors, and you get this amplification like an antenna. What is also very interesting and quite easy to understand is how this precise adaptation works. So as I mentioned, there's these enzymes, they add and remove these methyl groups, which is covalent modification of the receptors, which changes signaling. And you can write on some very simple equations, the rate of change of the methylation level. And then how it is, you can write a positive term and a negative term, one increasing, the other one decreasing the methylation level. And one is by a rate of methylation. And only the inactive receptors here are methylated. This is basically what we measure in FRET. And only the active ones are demethylated. And then you can immediately see how precise adaptation comes about. When things all settle down, the time derivative is 0, and you can solve for A, and the steady state value is a constant independent of any external conditions. So this is, of course, shown here. This A star value is always reached again. There's another way to think about a precise adaptation, which is very close to engineering, and this mechanism is also called integral feedback control. So there's a nice analogies with human engineering here. So when you want to design a chemostat, not a chemostat, when you want to track a thermostat, let's say an AC or an oven, you could organize it like that, so you add heat when you have an error in your temperature. Let's say it's too low, and now you want to add heat. However, that would lead to instability and oscillations because of lags, and some of the fluctuations are not real. So what is much better is you integrate over this error, and only if there's a net change, a net error, you add heat. This is a much more robust way to do that. And similar here in bacterial chemotaxis, this is also how it works. With the same model I described before, is then the amount of methyl groups added is the integral, proportional to the integral of the activity error. And that's also why before the areas have to add up to 0, because there was no net change in methylation, there was only a pulse which ended already, so the integral has to be 0. So now, I mentioned, you know, we have these pulses and step changes, but these are not these complex environments I talked about, and how can we infer about these? In reality, of course, there are many other cell types which change the environment of the cell I'm interested in. Let's say in an intestine, there are sources and sinks of nutrients constantly. And of course, the cell moves around and sees different things all the time. So instead of having one particular stimulus the cell might care about, there's actually a distribution of possible stimuli the cell might expect to see over its lifetime. And as this is described as this probability distribution. And it's not only one ligand, it's a high dimensional one with many different ligands and maybe also gradient and so on. The output distribution might be also, is also a distribution. It's maybe run and tumble, but intermediate values and so on. And so if we would know a lot about the chemotaxis pathway, we might be able to infer about the stimuli and the environments. For simplicity, and this work was done by 2 excellent PhD students before, one can simplify now the analysis as well by assuming the signaling. We don't worry about all the chemical details and dynamics. We just say the fast process, the receptors and motor are just described by these equilibrium input-output curves effectively. This is also quite close in spiritual engineering again. By the simplification now, you can make you can apply now a really nice theory again from engineering. It's information theory developed by Claude Shannon. So what is information theory in a nutshell on one slide, basically? Information, you could say, is a degree of surprise. So when you have a very flat distribution of potential inputs, so the cell has many choices it might encounter or many possibilities, this is a high entropy distribution. There are many possible things. It's all kinds of stuff. However, now when the cell sees one particular stimulus, then it's very informative for the cell. Among all these things which could have happened, one thing happens. However, if there's a very peak distribution and you get then your stimulus, the cell wouldn't be very surprised about it because it was expecting it. So that would be little information. You can also describe correlations, in particular between inputs and outputs of the cell by also information theory. It's called mutual information. And how does it work? So basically, if you have one information, what's the input, how much can I learn about the output? And if for all inputs, you always get the same output, there's very little correlation or mutual information. Here, I would have a very high correlation or mutual information. Positive, it could also be negative. So what can we do now with this? So here's a very simple slide, which gives sort of the idea of it. So to make progress on it, you have to somehow make first assumptions. You have to say the cells transmit a maximum of information. So it makes sense, this assumption, because otherwise why have an information processing or signaling pathway? Under these conditions, then you can derive relationships and can test them with experiments. So the idea was developed already early on by Simon Laughlin, and we applied it to chemoreceptors, in particular these 2 very talented students. So if you have now a distribution of inputs, what is the corresponding distribution of or input-output curve which maximizes this transmission? It's actually the integral, the integrative, the cumulative integral of this input distribution. So this is basically the matching one which matches the input distribution. What is the matching output distribution to here? It's a flat distribution, meaning you use all output levels equally with equal likelihood. So it makes also sense because you don't want output levels which you never utilize, which you never use, and that would be wasteful. And similarly, what happens here is the things you care about here, the stimuli are very highly resolved, these are very lowly resolved on the outside, which hardly ever occur. Again, also when you make this too steep, you would saturate too early. If you would make it too shallow, you don't utilize the outputs, and it would be wasteful. So that's a kind of analog computation. Of course, reality is much more complicated, but what sort of comes out of applying information theory to the chemotaxis pathway, sort of in a really nutshell here. Of course, we can now ask, how do cells would respond to different types of gradients, and they correspond to different environments effectively. What turns out is that the exponential gradients are the ones which are sensed best, either because really the cell cares about, or because they're very occurring very often in nature. And that also makes sense because cells are known to sense logarithmically. The internal representation is a logarithmic compression, and these are inverse functions, so you don't get saturation. On the sensing side, what comes out is that the information theory is consistent with contrast sensing. Only relative changes are important. And this is in line again with Weber's law from neuroscience. What does it mean? That the smallest detectable stimulus goes relative to the background. So when you're in a loud room, you all know this. People have to shout so you can understand them. If it's very quiet, people can whisper, and that is essentially what is happening here. You can also learn about motility. I mentioned briefly in the schematics that the motor is a very steep input-output curve, meaning if it's a step change, it's either on or off, and it's basically one bit. it's very little information transmission. However, we can resolve this by information theory by having multiple motors and by being conditional independent. This information flow from the receptors is conserved and it can be transmitted to the motors. So towards the end here, I wanted to come very briefly to this idea of, when do cells invest in this chemotaxis pathway, and is there some sort of cost-benefit trade-off the cell has to keep in mind? This brings me again to this little pie chart here, because the cell is finite, it has to make choices to invest in this or in this and so on. So just to understand how resources are invested to chemotaxis, we can just look, or the collaborator who does the experiment can look at the gene expression when a chemotaxis gene is expressed from some promoter, which is important. And what happens now is when he compares different nutrient sources, like bad versus good nutrients, meaning very low growth rate or high growth rate, or low fitness and high fitness, what is seen here, even without any gradients, when it doesn't grow very well, it invests in chemotaxis. So no gradients or anything. So it invests in chemotaxis motility, even if things are very bad, even if it's costly, effectively. However, now, so how can we learn about the cost and benefit and the fitness and so on? One thing to study fitness is a more robust way is to compare competitively the growth of, let's say, wild type, which has everything, and a mutant which is defect in something, and let's these grow together and compete and see who wins effectively. And then here, for instance, in this experiment, there's basically no benefit because there are no gradients, so there's no benefit of chemotaxis. And you compare wild type with a mutant which doesn't invest in the flagella, which is very costly. And you can see that wild type loses down here, and in particular when the growth rates, the nutrient is very bad. And this is expected, yeah. So this is a cost now of the fitness cost we quantify. However, now when you compare wild type with a different mutant, which is only impaired in signaling, and now there's no real cost difference, but there's a benefit because there's another bead which secretes ligand, chemicals, nutrients. Then we see when the cell doesn't grow well on the main nutrient, it can reach a new secondary nutrient and it has a fitness benefit. So we see really how chemotaxis here works. And so we have a fitness benefit to chemotaxis through this is additional nutrient when the poor nutrient sources conditions are otherwise prevalent. And yeah, so these lines here are also models, so we can describe these. This is a more complicated one here. It's a straight line effectively. So in summary, of course, I try to talk about a lot of things here. So I talked in the first part about the remarkable signaling problem, learn from these and the analogies to engineering and information theory. And in the second part, very briefly, I talked about recent work about the allocation of resources and that the cost is offset not by the direct immediate benefit, by the anticipated benefit. When things are bad, the cell is already investing in chemotaxis in the hope it comes useful. Yeah, so I wanted to, so what this really is all about is biological physics. You might also want to call it systems biology. And it's really an interesting area because there's a lot of people from very different backgrounds working in this area and coming together effectively. And in particular, it's a nice opportunity for people from a physics, math, and computing background to work on living systems, to apply their very quantitative methods and to work with some amazing biologists. And it's very rewarding. And I wanted to also acknowledge the students and collaborators and funding sources. So I think I stop here, and I'm really happy to answer your questions in the following session here we have. So thank you again for attending.
Well, thank you very much for the thought-provoking talk. And we will go straight into the Q&A session, and I would like to remind the audience that you can continue to post your question on the chat, and we will read out your questions if time allows. Right, so let's go to our questions already submitted, and again, I'm going by the number of thumbs up, and so the The question with the most thumbs up is a very interesting one. It's from David. And the question is, please, can you tell me how I would get into a similar field of study?
Okay, thank you very much. Thank you, David, for asking the question. So I mean, it's basically easy to answer, but the thing is, there are many different ways to get into this research area, and of course, it makes it exciting because, of course, many different people come together anyway. So even if you have a very different background, you can work on these problems. But if I would talk about myself and people like me or Chu Fan, one way is, of course, to study, in a sort of a very quantitative field like physics or bioengineering or even computer science or mathematics even, and then later on, let's say, after even your PhD or after your master's, at that moment to start collaborating and working on project in life sciences. But then there are also people coming from biology and say master's courses now, and I'm teaching on these as well, where you actually learn as a biologist also quantitative computational methods, and then you can apply these. And so you come from slightly different angles, but you sort of merge, and there are many ways to work in this area. So thank you again for this question.
Okay, right. So the next question is from a Dr. S.M.M. Young. I'm hoping for a comment on cellular communication as well, the signals they release into the environment for others to detect. So can you comment on the cellular communication?
Yeah, very good question. So, I mean, how I presented it now, I talked essentially about individual cells swimming around and sensing the environment, but I alluded, of course, in the beginning to the idea that these planktonic cells swimming around and so on is relatively, I would say, rare even. What is really happening is as they settle down and build these communities, which protects them, where they can specialize, they can specialize in particular areas or in labor and help each other. And how do they know now that they're in an environment which is very beneficial? And of course, that's where the signaling between cells comes in. What's the question about? What the question was about? And the keyword, I guess, is quorum sensing. So quorum means that cells can sense the amount which they have in the environment. And effectively, when all cells secretes this particular signal and it's not washed away immediately and so on, it accumulates and then the cells can sense this again. And at that moment, when a threshold is reached, certain genes are turned on, and then they, let's say, make a biofilm and extracellular matrix and so on. I mean, it's very complicated then, but we're also working on these kind of questions as well. Thank you.
All right. Okay, so the next question, what role, if any, has bioinformatics played in understanding how cells perceive their environments?
Bioinformatics. Interesting. It's interesting. Again, we come from all very different backgrounds. So I come from physics, and of course you can come with your favorite mathematical discipline as well and apply them. Of course, you can also come from bioinformatics, and that's actually how a lot of this quantitative biology developed very early on in the 80s, and physics came actually more later. I mean, how bioinformatics, I guess, I can only answer, I guess, more generically, I suppose. It allows you to, of course, when you sequence a new organism, to identify, what genes might be doing. Let's say you sequence something and you find there's most likely a gene, and then you run it against the database, and you see, oh yes, it's very similar or homologous to a particular chemoreceptor in E. coli, which we already sequenced and understood much earlier. You can say, there's a lot of sequence similarity, and now this must be a receptor. And next thing, of course, you have to figure out what it might sense and so on. But so maybe I stop here, but it's maybe how you would go about.
Right. Okay, so the next question is from Gian. How do cells adapt to the topological features of the surface they are in?
Very interesting. That's a very interesting question. I'm wondering now maybe if the question was more about the topography, sort of say, the surface height and so on, because that plays an important role. But let's stick with the topology for a second. It's not so easy enough for me to answer it in terms of bacteria, because a bacteria is essentially, quite a solid compartment with the cell wall and not so deformable and so on. But let's say if we talk about more broadly about eukaryotic cells, larger cells with a nucleus which can more bifurcate and send out protrusions and so on. That's certainly important because these cells can be in an extracellular matrix environment in our body, let's say, and this would be one topology. These fibers, they are interlinked, and how cells might respond to this sort of topology might be by adhering and pulling themselves, and that would be mesenchymal migration. But it could also have pressure-driven migration by blebs, and it's called amoeboid, which you can also do on a flat surface, which is in a different topology. If it's about topography, sort of the high profile, I mean, the single cells can settle down, and especially when you have fluid flow and so on, of course, there's no fluid near the surface. And depending on the topography, it might then settle down in these little cavities and more easily spread out and form a biofilm. So indirectly, it will benefit from it because the flow profile is different. So I think that's maybe how I can answer it sort of in quickly. So.
All right. Okay. Thank you. So the next question is from Charlotte. Can engineers use biomimicry of how cells perceive their environments to improve mechanical processes or computer algorithms? And how can mechanical engineers get involved in biological research?
Well, this was a very elaborate compound question. So I took a little bit a note here. So the first one was about the biomimicry, how it can improve mechanical processes. So that's probably the hardest part. Maybe I start with that. It's not fully clear how to answer it so quickly, but earlier on I worked on a problem in sporulation. So meaning, again, about bacteria, of course, when bacteria grows, they elongate and divide and elongate and divide and so on. In sporulation, you form an asymmetric compartment, which is later remodeled into a spore. But what I wanted to basically say is when I gave a talk back then about this topic, I compared a cell in the remodeling and how hard it is, which is under high turgor pressure. Yeah, it's super high pressure. And that's why you need a cell wall. And this remodeling is very difficult because if you imagine it as engineering, let's say you have a submarine deep down in the water and the pressure is now coming from the outside. And if you wanted to now, mimic cells, then you would be able to repair or even divide a submarine underwater, deep water. And that would be a very challenging engineering feat. So we can certainly, hopefully, learn a lot from bacteria. I don't know how to make a submarine like that, but that would be one way. And then the second one was about the computer algorithms, how it can, I guess, be used to improve computer algorithms. I guess there's a lot already known about that because, bacteria try to optimize the environment or swim towards optimal conditions by chemotaxis. You could compare this with search algorithms or optimization algorithms we have already. I mean, there's stochastic gradient descent, where you basically optimize stochastically in certain directions you want to go, and you optimize some function, which is similar to chemotaxis. Also, of course, in neural networks are simulating effectively cells. So in neural networks like deep learning, you have activation functions, which are sigmoidal, and similar in chemotaxis, we have these sigmoidal signaling functions. So in neurons, you have maybe that represents membrane potential. In our case, it's signaling. But so that already informs current algorithms. And maybe one other thing about it, I mean, I talked briefly about the output distribution of being very flat. So this is already used in digital image processing. So it's called histogram equalization. So you basically try to have some image with a very narrow distribution of intensities, which is very too light or too dark. You spread it out over all the potential channels you are allowed, and then you increase the contrast. So what would be another way? The other question was about the the last one was about how mechanical engineers can get involved.
Mm-hmm.
I mean, mechanical engineers are very much involved already. There's an area in cell biology and tissue biology, which is called rheology, where you perturb stretched tissues, similar how you might study fluids, viscous fluids. And it's a very hot research area actually right now. Of course, mechanical engineers, bioengineers, fluid mechanics people say design flow chambers where you study stuff. So there's a lot of equipment also, which is very much related to mechanical engineering. So yeah, good question. Very, very broad question.
Right. Okay, so the next question is, do we have data and information on how cancer cells perceive their environment to grow and reach metastatic niches?
Yeah,
so
it goes into the so what I talked about today was the chemical sensing and adaptation and how cells respond. I mean, this question is presumably more about mechanosensing. So so I guess it's something which would be related, not chemotaxis, more to durotaxis, the stiffness sensing of the substrate, maybe haptotaxis, also sort of ligands immobilized. Anyway, how do they sense? What was the question? How do they sense the environment?
The question is, do we have data or information, or maybe just how much do we know about cancer cells finding the metastatic niches?
Yeah. I mean, I don't work in this area specifically, but of course it's a very hot area, and of course I heard a lot of talks about it. So yes, the mechanical properties of the extracellular matrix are very important. Tumor environments are very special in the chemical environment, in the physical environment, and I guess, you know, even sort of internal pressure of these tumors is very important. And when some sort of threshold value might be reached, cells, the mobility kicks in and then they move out. So I think generally can't answer it so easily, so quickly, but I think the mechanical, it's very much known the mechanical properties are super important for metastasis. So cells have to squeeze themselves through pores. It's certainly super important, yeah.
Okay, yeah. Thank you. So the next question is from John. How are mechanical forces measured at the cell level? Is it similar to the tissue level measurements made for chemical and electric gradients? So how are mechanical forces measured at the cell level?
So I mean, I guess I would need some more information, but I guess there are two ways. Is it about the experimentalists trying to measure things at the cellular level forces, or is it the cell sensing it? I mean, I guess quickly answer both ways. So of course, if you're an experimentalist and you want to measure something at a cellular level, I guess there are sort of techniques like atomic force microscopy, you know, where you sort of poke the cell and based on the pressure, you can see how you learn about the material. I guess there's something like traction microscopy where you have beads in the substrate and the cell attaches to it. I guess I'm talking about larger eukaryotic cells, and they attach, adhere, and contract. And then you see the beads deform, and you can look at this under a microscope with sufficient contrast. There are optical tweezers where you can manipulate single cells and poke them and understand the forces. So, but how cells are measuring it, I guess I talked about the chemoreceptors, which unbind ligand. In this case, you also have receptors. I mean, there's adhesion receptors, which under tension, let's say, unbind less strongly. They deform, show more binding sites. They become stronger. So normally, when you pull something like under tensile forces, you more quickly unbind. These are slip bonds. But when you have a catch bond, like an adhesion receptor, they get stronger. And this is basically a mechanical sensor. Another one which comes to mind in bacteria is mechanosensing ion channels. I think they sit in the membrane of the cell, and when there's increase in Turgor pressure, the thing swells and gets expanded. Membrane gets thinner. The channel changes conformation, it opens up anyway because of the tension, and it lets out the fluid and compensates for that. So these are two molecules which are force sensors.
Yeah, Okay, so there's a second part to the question, is whether it is similar to the tissue level measurements made for chemical and electric gradients. So I guess it's whether at the cellular level the measurement done is similar to the tissue level. That's the second part. Mm-hmm.
Okay. Yeah, I don't know about the electrical measurements. I don't know, but let's stick with the mechanical stuff for a second, which may be easier to answer. I think, you know, we just last week or 2 weeks ago in our Physics of Life seminar series, we had Margaret Gardell from University of Chicago, and she gave a really nice talk. She talked about these epithelial cells, these cell layers, which are in our intestines as well, and so on, of course, skin. And effectively, what can happen is under mechanical stimulation is that, again, the adhesion receptors between these cells, the tile, and the adhesion receptors, the tight junctions, and they can get stronger, and they recruit actin, they form bundles and myosins, and they can contract and become stiffer in response to environmental changes. So ultimately, it's again starting with adhesion receptors, but then, you know, there's all kinds of stuff recruited, and the signaling cascade is essentially exploding and doing all kinds of stuff. But yeah, it starts with that, and then, yeah, I think that's mainly my answer.
Okay, yeah, thank you. So the next question is, how do sublethal concentrations of antibiotics affect chemotaxis in bacteria? So what happens if we put sublethal sort of toxins in the bath, and how would it affect chemotaxis? It's
a good question. If I would think more about it, I would probably come up with some good answer, but it's good question. Let me just think. I would
certainly credit to our audience asking thought-provoking questions.
Good question. I mean, of course, I talked about attractants like nutrients, where they move towards the cells. Of course, there are a lot of repellents as well as the cell tries to avoid. These are metal ions. There's all kinds of other stuff. I guess antibiotics, I don't know if they sense by chemoreceptors, maybe not.
Would it destroy part of it? I mean, would antibiotics destroy some of the sensing ability, or is much more generic, just destroy the membranes?
Yeah, I mean, of course, antibiotics, they attack the membrane biosynthesis. They attack also, I think, the function of ribosomes. They're kind of, you know, of course, lethal. So also attacks the biosynthesis of the cell wall, the peptidoglycan layer. How this now interferes with chemotaxis, I can't really answer it very well immediately. But of course, once you have a really strong stress response, again, you know, resource allocation might come in, and it might actually divert some of the resources to other things. So and try to repair this problem. So yeah, good question. Really good challenging question.
Thank you. So the next question is, how would you suggest natural selection has been, if it has, involved in creating these cell signaling systems?
No, that's also a very good question. I mean, I like this question because I like the questions, the interface between very simple physics intuition and explanations, and then of course we deal with biology, so now you have to deal with all this complexity which evolves over millions of years or billions of years in terms of bacteria, and then you end up with these very elaborate signaling pathways. So at some point I wrote a paper about it a few years ago, about adaptation, how it could have evolved in protocells. And it's basically, we basically assumed, you know, you have a receptor in the membrane, a protein. It doesn't even have to be some fancy receptor, which is in 2 conformations and so on. And it senses something on the outside, it binds. But then how would it adapt without having some really elaborate signaling pathway, like in E. coli and so on? And one way would be that you have actually not a perfect membrane, things are diffusing in and out. And then ultimately, you know, even if you're a cell and you're sitting all of a sudden in a high ligand concentration, the receptors changing potentially, but then it leaks also inside the cell and there's, let's say, a second binding site on the inside, which restores it to conformation to the original one, and effectively the receptor is adapted. So, I mean, I like the idea that there's very simple mechanism, like a two-state receptor, and then, you know, biology adds this complication and regulation and makes it sort of optimal. But the core, I believe, is a very simple physical mechanism. So, and it's always fascinating to understand where the physics came in and what else is added on. So it's a really nice question. Thank you.
Okay. So the next question is from Sarah. Cells have perception. So that's the question. Do cells have perception?
I would argue yes. So, I mean, what is perception, right? I mean, what is our perception? You know, we have stuff happening in the environment, and then, you know, we have a retina with photoreceptors and an optical nerve and layers of complexity, and everything ends up in the visual cortex after filtering and hierarchical coarse graining. This is, of course, what's imitated in convolutional neural networks. So, I mean, what is it? You know, it's just electrical signals, and we then have the illusion of seeing a picture in front of our eyes. You know, it's basically what is perception? It's an internal representation of the environment inside us or inside a cell. I guess what is inside an E. coli cell? I guess it's it is basically the activity of the receptors, which would be the first internal representation. We actually talked about this in some papers. So there's some free energy of the receptor, which determines the switching between the two activity states. And this free energy has some logarithmic dependence on the ligand concentration. I could go into it. So this would be the closest to the first perception of the external environment. It's a compression, and the logarithmic dependence is because of the chemical potential, and it's basically in line with Weber's law. So the logarithmic dependence is actually the integrated version of the Weber's law, which is called Weber-Fechner law. And that would be what I would call personally perception of chemotaxis in E. coli. A very cool question. Thank you.
Yep. The next question is from Jamila. Can cell perception be used for smart materials? For example, touch screens, etc. Can we use cell perception to do things, to make smart materials?
Oh wow, yeah, wow. I don't really know very well. I would have to first think about how these smart materials really work, and I guess I'm more on the fundamental physics side in biology. So I maybe pass this question, and if anyone wants to ask me personally in email, you know, drop me an email.
Yeah. Well, okay. So the next question is from Vinita. What would be the one thing that you would want someone to take away from this presentation?
What is it? One thing? I mean, what is it in a nutshell about? It's really said. I think I showed you this one slide with all this complexity, right? And the biochemical, metabolic pathways and gene regulation and whatever. I mean, and you know, we like to think that things are modular and independent of other things, but you know, in reality, it's all context dependent. And people who work in synthetic biology realize, you know, this problem of putting pathways in other cells and this context dependence. So what I guess what I would like you to take out of it, from this talk, is that you know that you can have minimalistic modeling and a very, I think, also quite deep understanding of complex biology reduced to very simple principles. You know, you can abstract the pathway into just a couple of variables, very simple things, and you can even uncover the remarkable signaling properties, and it has a huge explanatory power. So I think the simplicity is a take-home message, and I think that's also where physics really contributes and complements biologists who like to embrace the complexities. This dualism is, I think, really important and very fruitful, actually. Very cool question.
Okay, great. So the next question is from Azim Khan. I read that how cells sense that they need to divide. It said that the difference in the volume is a factor for sensing the timing for division. It will be great if you can explain how cells can sense this small volume.
Yeah, I mean, I know it's a very hot research area. I'm not fully up to date with this research area. Of course, if a cell grows and it makes more proteins while it grows and the volume increases, the concentration is the same, and it's not so fully trivial how to measure the volume of a cell. I mean, maybe I deflect a little bit and talk about the MIN system for a second. Maybe that wasn't fully what the question was about. So how does the cell find the middle? And it has also to do with the length, of course. So the cell is growing, and there's a particular very small pathway, again, called MIN system, which tends to bind to the cell poles, and then it recruits a molecule which removes it again. And then when it's removed, it tends to attach to the opposite cell pole. And what it happens is then this is basically an oscillator inside a cell. So it goes back and forth, and it's very similar to Turing patterns as well, actually. So, and this dynamic system effectively has the effect that when the cell grows and it shuttles back the proteins, they have a little bit more tendency to be at the cell poles, and there's a lower concentration in the middle. And it's this low concentration which acts as an inhibitor. You are basically then forming a FTZ ring, and that recruits the machinery to divide the cell and pinch it off. And that does depend on the cell length as well, because these oscillations are only develop at a certain cell length, and that's how it kickstarts. So, but it's not fully addressing the question of volume. It's more like cell length, but the diameter is more or less fixed in an E. coli. So length is equivalent to volume in some sense.
Yeah, right.
Question.
Okay, so it's coming to the end of the event. I will ask one last question. That's the highest voted question from Laurie. Even though they are not really cells on their own, but need to infect a host cell, I would like to know about viruses. Do the variants have the same kind of processes to get a sense of their environment when they are not yet in a host cell? And if yes, does it play a role in the infection process? So can viruses sense?
So is the question about viruses, or what's more the knowledge of viruses in bacteria?
So it's about viruses. Can viruses perform any of the sensory adaptation or sensing that you described? And how would that affect the infection process?
I mean, of course, now I could talk about bacteria. Of course, bacteria sense environments, they have the receptors and so on. About viruses, I mean, I basically take a very quick shortcut to answer this question. I mean, as we all know, viruses are not alive. No, I don't want to just stop here, but basically I'm saying is there's a capsid, and it's a quite an inert capsid, effectively material. And as far as I know, you have of course the DNA or RNA content, and of course there's no active sensing of the environment. I'm pretty sure about that. Of course, there's indirect sensing, right? Because similar to spores, let's say, and I worked on this a little bit, if they're in the right environment, and I'm sure a virus capsid with a load in it is in the right environment, and the enzymes are there, let's say, if it's in our intestines or in the saliva, I'm sure there's these enzymes which then actually, on accident, basically digest the hull and release the cargo. And it's similar with spores. So if the humidity is high, then they inflate the water goes in, and it inflates and breaks the shell. So indirectly, of course, the material is designed in a smart way by evolution to sense. But I don't think there's any active thing because it's a dead object. But yeah, very, very cool current question.
Okay, fantastic. So I would like to thank Robert again for the really fantastic talk, and I would like to thank the audience for the really intellectually stimulating questions. And I think this really highlights the field of biological physics. It is really an interplay of many different scientists and engineers and researchers. And I would say, personally, it's the most exciting field of emerging science at the moment. But so I would like to thank everyone again for joining us for this Science Breaks event. And please join us for the next Science Breaks event taking place on the 8th of October to hear Professor Gareth Collins talking about the dinosaur dooming asteroid. Thank you very much.